We are going to go ahead and get started with some introductions. Thank you for coming to our first annual, hopefully, BMES Design Competition. So the goal of this competition was a number of things. One, to, as a society, start to participate in and recognize the designs that are coming out of our group, and to actually see those in action. For students to come and be able to present on design projects, which is not something that's typically done at conference venues, but to both engage the audience and to really disseminate the practice of design within our disciplines and present that to our peers. I'd like to thank our sponsors. This design competition has been sponsored by Medtronic, and thus the theme of this year is bioinstrumentation. With the diversity of topics and content in BMES, it was our hope that we would have a rotating list of concentrations in the design competition. So this year is bioinstrumentation, and we hope to cycle through biomaterials and biomechanics in the coming years. I'd also like to thank the BMES leadership for allowing us to do this, an entire session, and a lot of legwork to get this taken care of, and then our judges, our judging panel, for volunteering their time to evaluate. And most importantly, the design teams for submitting. So this year we had 16 entries, and we whittled those down to top 6, and you are here today to present 4 prizes. So first place will get $3,000, and second place, I think, is, how much is it, Liz, second place? $1,500, and then $500 for third place. So real money in the next hour and a half. We'll see how it happens. Without further delay, we will have our first team. Just to let you know the rules, there's 10 minutes for presentations, 5 minutes for questions. We can have questions from anybody in the audience, so here's your chance to, see what great questions you can come up with. And our first, we have University of California, Berkeley, Columbia University, Yale University, Worcester Polytechnic Institute, Virginia Tech University, and Virginia Commonwealth University. So our first speakers will be from the University of California, Berkeley.
All right, there we go. Hi everyone, my name is Neil.
And I'm Lucy.
And we're here on behalf of UC Berkeley, and we're going to talk to you guys about this project we've been working on called the self-cleaning laparoscope. But before we talk about that, we want to talk about what laparoscopic surgery is in the first place. So laparoscopic surgery is a form of minimally invasive surgery in which the surgeon basically makes incisions onto the patient's abdomen, and they insert ports. If you look at the video playing, we insert ports and basically have trocars as well that kind of, as you can see, there's a trocars being placed right here. Basically, the main purpose of this is to basically create a surgical area without actually having to do an open procedure, which is basically cut open the entire abdomen and view the entire space. You insert instruments and cameras, like as you can see here. Basically, the idea is you perform the entire procedure using a camera, a laparoscopic camera, and you have instruments that basically cauterize or ligate and even resect different parts of the tissue as well. So one of the benefits about laparoscopic surgery is that you minimize patient complications as well as you increase the rate of basically patients returning back to their normal activities. You also decrease hospitalizations by a significant amount as well. Laparoscopic surgery is definitely a growing field. Basically, it's $3.5 billion scheduled, it's its predicted value by 2017, and there's currently 35 million laparoscopic surgeries being performed in the US currently. And also, there's an increasing number of minimally invasive surgical fellowships in the US. There's currently 156, and this just goes to show that basically laparoscopic surgery is a field that's continuously growing, and it's gonna be going in the coming years as well. So, what's the unmet need? Basically, the idea is you're putting this camera, this very thin camera, into a port into the patient's body. And so the problems with that is that sometimes there's tissue smearing, like the tissue debris from the patient can actually adhere onto the lens, and this will distort the vision. There's also abdominal bleeding. If there's a particularly bloody procedure, any kind of blood that goes onto the distal lens itself will basically occlude vision. And finally, there's a temperature differential between the patient's body as well as the surgical OR. So any of these situations can basically occlude the vision, and the surgeon's basically performing a procedure blind. So the standard of practice is what they do is they basically physically remove the laparoscope, clean it off, and put it back in. And this could take up to, even more so, 30 minutes in a 4-hour procedure. As you can imagine, performing a surgery when there's abdominal bleeding can be pretty risky, and it's potentially life-threatening as well. And this is a very frustrating aspect for surgeons as well, because the surgeries that I sat through, they would often remove it several times, and it would just prevent them from actually getting the work they needed done. So to address this unmet need, we looked to what was actually on the market itself. We looked at several different companies, including Stryker and Olympus, which make laparoscopes, and we looked at several different parameters, including the diameter, the shape of the lens, the resolution, et cetera. The two things that we wanted to focus on the most was basically a cleaning count. We wanted to make sure that we can create a device that will decrease the amount of time needed to clean during the surgery, and we wanted to make sure that the resolution was adequate. So as you can see on the right, that's our ideal values that we use, basically comparing our ideal product to what's currently on the market. So what's our idea? The idea is very simple. It's actually based off a ballpoint pen. So the way the ballpoint pen works is you basically have a ball that sits at the end of the laparoscope and at the end of the pen, and as soon as you put it on a piece of paper, basically the ball gets dislodged into the pen itself and ink starts flowing. What we want to do is instead of putting ink out, we want to take debris in. So it's basically, imagine a ballpoint pen, but in the opposite function, I guess you could say. So here's our idea. Basically, you have on the right, so you can see here. On the right, you basically, on the left rather, you have this glass ball that's encasing this lens right here. And this ball is powered by a motor. This motor is controlled by a button that the surgeon has access to at his fingertips. So when there's any kind of debris that latches onto the laparoscope itself, the surgeon can press a button which will call this motor to rotate. And this ball will basically start rotating. And there's a rubber wiper which will basically prevent any kind of macroscopic debris from actually entering the laparoscope. But any kind of tissue smudging or any kind of bleeding will actually go into the laparoscope itself. And there's an integrated saline and vacuum channel which will basically clear off this kind of imaging lens. So the point of this whole design is basically to make sure that there is a clear optical pathway during the entire procedure without having to physically remove the laparoscope itself. And as you can see here, this is basically just the front end of the view. You basically have the glass ball that's rotating this way, and there's a lens that's hidden and sitting in the middle over there. And so the reason we came up with this idea in particular was because we talked to several surgeons. We tried to figure out exactly what they wanted. The things that they wanted the most were ease of cleaning and clarity of vision, which is something that we rated pretty highly on our scale. And this device is actually satisfies both of these criteria because you are continuously cleaning during the surgery, and the lens is protected by a glass sphere inside. It's also other parameters we identified was ease of use, portability, ergonomics, and functionality, which we think our device does pretty well.
So to prototype our design, first we modeled it in SolidWorks and then 3D printed a looks-like model, and we built a large-scale works-like model. So we conducted 2 tests with our prototype, resolution testing and cleaning performance. With resolution testing, we wanted to know what is the effect of putting a lens inside a glass sphere and what are the optical effects on that. And then for cleaning performance, we want to determine the efficacy of this proposed ballpoint pen method. Does it actually clean? So the results of our resolution testing, we have the resolution target without the glass sphere and with the glass sphere. You can see that with the glass sphere, it does introduce a lot of aberrations optically. It does look very fuzzy. However, this is done with a large stock glass sphere available to us with a high precision manufactured small 2-scale lens. We could minimize these aberrations. You could further minimize aberrations by optically adjusting the other lenses in the laparoscope. Despite the fact that it's fuzzy, the resolution doesn't change that much. The minimum resolution you can resolve is approximately 5 microns without the glass sphere. It only increases to approximately 6.21 microns with the introduction of the glass sphere. So even though the image does look fuzzy, you can still resolve very small optical factors. So to prototype our cleaning performance, we did a water vacuum combination on the outside to mimic the internal cleaning system, and then we had the motor running on the inside. And we used jam as substitute debris, and our cleaning time averaged 3 to 5 seconds. We had to run the water and vacuum on the outside because our facilities didn't have the capability to watertight seal our internal electronics from the water that we were pumping in. So the takeaway from this chart is pretty much our prototype, we managed to get our desired value from our cleaning time, which was the need we were focusing on. We minimized the amount of cleaning time necessary for that, while still maintaining other aspects of the laparoscope within parameters suitable for use, because you couldn't compromise so much on other things just for cleaning time. And so our prototype has some limitations. The first one being it's scaled up 10 times from the actual proposed device, and we had a lack of saline and vacuum channel integration. Beyond that, our design has some limitations itself. We can't just simply put an add-on to the end of a laparoscope because you have to integrate the lens inside this glass sphere. So you would have to create an entire modified laparoscope. It's mechanically complex, which provides room for malfunction, and there's a clear trade-off between image resolution and cleaning time. Economically, the main difference between this and a standard laparoscope is the integration of the sapphire glass hollow sphere. And so we would need two 10mm sapphire glass optical domes. They're pretty much hemispheres, and you fuse them together to get your full hollow sphere. These are already mass manufactured by optical lens companies, so there's no cost in infrastructure for us to manufacture these. However, the material itself is expensive. This entire sphere could cost up to $400, and the integration of the other aspects, millimeter scale motor, O-ring, lens holder, 2 lens posts, that's negligible in terms of the cost of an overall laparoscope. If we add in the cost of propose an additional $100 per laparoscope for manufacturing, that brings our standard laparoscope price from $1,700 to a proposed $2,200. However, this increase in $500 is justifiable because, one, this is a reusable device. This increase in cost gets spread over multiple patients. But also, the saved time in the OR in terms of cleaning will easily make up for this increase in cost. And so our design has actually been recognized by the industry. We're recognized in outpatient surgery. We've submitted a provisional and a non-provisional patent and are currently waiting for the results of that examination. Our future plans are to refine the prototype with medical design firms and to contact laparoscope manufacturers that would be interested in integrating this technology into their current devices. So thank you. Any questions?
If you have any questions, please come up to the microphone in the back.
Sure.
I had a question.
Sure.
So how might you deal with the potential for contamination of the laparoscope that you've designed, given that it does have exchange of fluid and vacuum across the membrane?
Right, and that's a good question. So I think one of the big things that we wanted to also address is that basically while one end of the laparoscope has this ball, the other end is completely clear. So basically you can actually use a autoclave machine to actually clean it off as well. So that's one aspect. And a lot of surgeries, they actually have on-site specialists who are from the device company too. So they can probably help with the cleaning process if there's a problem during the OR, during the procedure itself. Mm-hmm.
Hi, this is a great idea because saving time in surgery is really important.
Thank you.
My question is, how confident are you that you can scale your prototype down to a feasible size to really do laparoscopic surgery?
Right, and that's a great question. So laparoscopic surgery, like the lens itself is on the order of like centimeters to even millimeters, right? So we looked at the design that we made, the initial SolidWorks design, was actually on that millimeter scale, so we know that's functionally feasible. And the materials that we used and we researched, including millimeter motors and things like that too, those are also marketable and available online and from different companies as well. So we know that the different parts are available. It's just the next step is just a matter of putting them all together and to develop that one laparoscope that has all the features, I guess.
Right, thanks.
Mm-hmm.
My question's in terms of your economic plan. So you say that the cost of materials are really, really expensive. So I'm just concerned that it's just not the cost of materials, but also if you're adding $500 now, if you're trying to market this, that adds, you need to make a profit on that $500, right? You
Right.
need to make 4 or 5 times profit to pay for engineering, et cetera. So my question is, how can you justify paying probably upwards of $5,000 for a laparoscope just to save, a short amount of time, almost double or triple the price?
So the proposed increase is actually based off of what current laparoscopes sell off of. So the current manufacturing costs and material costs of a normal laparoscope aren't $1,700. It's much lower than that. So ours is really an increase of, let's say you still make the same net profit as a standard laparoscope, you just add in the addition of an additional manufacturing process, so only that gets increased. Foreseeably, you could even market this up to $3,000, but the main issue is that's a marketing issue at the end of the day, but foreseeably, you don't need to sell this at $5,000. The only increase is going to be approximately $500. So if you want to maintain the same net profit as you get while selling standard laparoscopes, like the Stryker and the Olympus ones, you could foreseeably do that with just a higher end model.
And I think one other thing I want to add, and to your question as well, is basically the point is, the idea we're using here is time equals money. So basically the 30 minutes you save from a procedure, a surgeon does several procedures throughout the day. You basically can do more procedures, which will help pay for the laparoscope too. And because this is a one-time cost, you're buying an entirely new laparoscope, that money will already make itself worth its while. Like, you'll get more money, more dividends, because you bought this one tool that's saving you money day after day after day. So it's a little bit more of an initial investment, but you'll save a lot more money in the long run.
All right, that's all the time we have. Thank you very much.
Thank you.
Thank you.
So next up, we have Columbia University.
All right, good evening, everyone. Good afternoon. My name is Sean Thomas, and I'll be presenting today on behalf of the rest of the team at Columbia University. So today I'd like to introduce you to our device, the HydraSystent, which is a military-specific solution for combating dehydration. So first I'd like to walk you through a bit more of the background on the problem. So dehydration, as most of you probably already know, occurs when the body loses more water than it takes in. And unfortunately, military personnel tend to be at high risk for dehydration, not only because they tend to be deployed in hot and dry climates, which accelerate the rate of dehydration, but also because they're carrying a lot of gear and equipment with them, which makes them sweat more and they lose a lot more water that way as well. On top of that, they only can carry a limited amount of water with them out into the field, and so they have to ration what water they do have in an intelligent way. So just to give you a sense of how big this problem really is, for the last decade or so, we've had over 400,000 troops deployed in the Middle East, putting them at high risk for dehydration. Over the summer, temperatures in Baghdad and Afghanistan can get as high as 120 degrees Fahrenheit for weeks at a time. And if you can imagine, you can take a look at some of the symptoms we have up on the screen. If someone had a hallucination in the middle of a field mission, it would have disastrous consequences for both the soldier and their team. From what we've gleaned from our interviews with military personnel, there's really no good way to assess whether or not a person is dehydrated, and if they are, how severe the dehydration is. And so these injuries tend to sneak up on people, and when they do hit, they slow down the entire unit because they can't just leave someone behind. So what they really need is an objective way to tell whether or not they're getting dehydrated and how severe this dehydration is. So if you take a look at the image on the right, you can see pictures of military personnel, and they're clearly carrying a lot of gear with them. It can be in excess of 50 or 60 pounds sometimes. And so from what we've heard firsthand in interviews with them, they've told us that if a device is too complicated or gets in the way of what they're doing at a given moment, they're simply not going to use it. And so keeping that in mind, we came up with a few requirements that a device would need to have to be successful in this kind of environment. So with that in mind, I'd like to sort of walk you through the design for our device. So our device is integrated into the standard Camelback hydration pack that's given to every military person who's recruited into the Army. And so on one end of this hydration pack, we have the actual spout where the person drinks water out of. And on the other end, we have our sensor, which is a saliva conductivity meter. And so the way this works is that the user can deposit a saliva sample into one end of the device. The processor analyzes the conductivity of the saliva, and then a number of LED lights will light up on the panel based on how severe the dehydration is. It's a very familiar design for people who are in the military and have used these Camelbacks before. The processing happens pretty much immediately, and the LED readout is simple enough for pretty much anyone to understand. So I'm sure a lot of you must be wondering, what does saliva have to do with dehydration? So this is a figure from a paper that was published a couple of years ago that correlates the severity of a person's dehydration with the osmolarity of their saliva. And so you can see that as a person gets more dehydrated, as they start losing more water from their body due to dehydration, the osmolarity of their saliva increases. Unfortunately, saliva osmolarity isn't something that can be easily measured using a portable mechanism like the one we want to use. So we tried to find some way to measure that indirectly. And that's how we settled on conductivity. Because where ionic solutions are concerned, conductivity and osmolarity are highly correlated. And saliva, as it turns out, is pretty much just water with a few ions and other biological components. So to see whether or not our device could reliably track these changes in osmolarity, we created a few different simulated saliva solutions based on a recipe we found in the literature. And so we made these at different concentrations to simulate varying levels of dehydration. And so you can see in the graph that the conductivities that our device measured correlated quite nicely with the osmolarities of the solutions that we made. And so the R-squared value is over 99%. But it's not totally unexpected to see this kind of trend because it's pretty much a physical law that as concentration increases, the conductivity should increase as well. The real test would be to see whether or not our device could reliably track changes in dehydration state in real human saliva samples. So we asked a few volunteers, namely our professors and TAs, to go to the gym and collect a few saliva samples for us before and after their workout. And we also asked them to keep track of their weight before and after the workout as well. And so you can see for most of the subjects, there's a very distinct clustering in the pre- and post-workout saliva samples. So the blue dots that you're seeing on the screen are pre-workout samples, and those are at a lower conductivity and osmolarity. And that's because the person is more well hydrated, and so they have more water to put into their saliva when their body makes it. And so the post-workout saliva samples are at a higher conductivity and osmolarity, and that's because their saliva is getting dehydrated as they're getting dehydrated. So we should note that not all of the subjects produced the trend that we expected. So you can see subject 4, the clustering isn't quite as distinct, but we're confident that with a more rigorous clinical protocol and adding more subjects into the study, we can get a lot more power to these results. So overall, the results were pretty promising, and we're ready to start distributing this product to our users. Now, we never claimed to have built the first dehydration sensor there ever was. There definitely are other dehydration sensors out there, but ours is the first one specifically tailored to the needs of military personnel. And I also would like to mention that the method that we're using to do this, saliva conductivity measurements, has never been done before. And along with filing for a patent, we're also going to try and publish a research paper for the scientific community, just because this has never been studied or researched before. So just to give you a bit more information about the market that we're looking at, we're talking about the 700,000 active personnel in the military right now. And that's not even counting the other markets that we could potentially expand this technology to. The United States is interested in acquiring technologies that can help their military. And so we're confident that we can convince them of the need for our device to help their soldiers out in the field. Speaking of cost, our prototype cost about $100 to build for this unit that I just showed you. That's actually a bit of an overestimate. And we're confident that as we scale up production, that cost will come down. And this is kind of a rough number, but say we sold these at about $200 per unit, we'd be sitting pretty nice with a nice profit margin. Now, $200 might sound like a lot to pay per unit, but the price per unit might not matter as much as you think. It's true, the users that we're targeting are the Army and Marine Corps, but the actual buyer of the product is the Department of Defense. And so if we can convince them to get on board with this, they'll buy this product in bulk for every military personnel that's in the Army now and in the future. You can think of it as we're creating a new industry standard in the military, guaranteeing us sales both now and in the future. So moving forward, we'd like to clean up the design of the device a little bit to make it more elegant and easy to use for the customer. I know some of you are scratching your heads at the idea of spitting into a collector cup. We can definitely do better than that. And so this design that you're looking at on the PowerPoint allows the user to put the device directly in their mouth to take a measurement, which is much easier to use. And ease of use is really going to be the key to getting this product to take hold in the military. So we'd like to distribute this product to the military, obviously. And to do that, we're going to try and partner with Camelback and license our technology to them to use their production lines to get our product out to our users. Camelback already has a great relationship with the military. Everyone is issued a standard Camelback hydration pack when they join. And so we're confident that we can convince them to create a new line of products centered around our device. So after monitoring the performance of our product in the military settings, we'd like to expand this to new markets, namely collegiate and professional athletics. So I won't dive too deep into the details of this, other than to say that athletes have a very huge need to monitor the intake of fluid that they have, not only to prevent dehydration, but also to prevent overhydration, because that can seriously affect their performance on the court, on the field, wherever. So this is a timeline of what we've got planned over the next few years. I know there's a lot going on, but I'd like to point you out to a few specific milestones, namely our filing for a patent and a 510K regulatory approval from the FDA by the end of the year. We'd like to start talking with Camelback by the beginning of next year to try and get shipment of this product out by the end of next year in 2016. And then based on how that goes, we can start shipment of the athletic line in 2017 and beyond. I know it looks like a really ambitious timeline, and there's still a lot of work to be done, but we're really confident in the technology that we've built and for the need that, and for the potential that it has to help people in the military and in other markets. Because at the end of the day, that's really what it's about, right? Helping other people. On that note, I would be remiss not to acknowledge the people that have helped us along the way. First, we have Dr. Aaron Kyle in the audience, who's been a close advisor with us since the inception of this product. And also Drs. Barclay Morrison and Elizabeth Olson, who helped us a lot with the early experimentation, and the MIT Lincoln Laboratory, who presented us with this issue to begin with. All right, thank you all for your time.
Hey, great idea. Couple comments.
Sure.
So 700,000 military are not in the Middle East, right? That's not, is that the full market of?
Well, no, you're right. So 700,000 people aren't in the military, but every person that joins the Army is given the same combat outfit,
Yeah.
along with some modifications. People who go to who are actually deployed overseas will have a few other components, but certain things are standard, including the Camelback hydration pack.
And then I wondered, does having a TA work out for an hour and spit in the cup simulate sitting in 115-degree weather
Yeah, no.
quietly in the full battle fatigues that cause hydration?
No, I mean,
It
you're
may
absolutely.
not be an exercise thing.
Yeah, no, you're absolutely right. I mean, the protocol that we use to get saliva samples and simulate dehydration, it's definitely in its early stages. And that's also why I mentioned that we'd like to develop a more rigorous clinical protocol. The people who we had to work out for us maybe only worked out for like an hour at a time. But I mean, on that note, it's promising to see that we still saw that clustering despite how not rigorous those exercises were compared to what people in the military would see. So once we have like an actual protocol going, we'll probably have them exercise for a longer time, standardize how saliva samples are collected, and that way we can get even more power to these results.
So thank you very much. That was a very good presentation.
Thank you.
I just have a comment. My mom's a nurse at the University of Michigan Hospital, and she takes care of a lot of patients who they don't know if the patients are dehydrated. And what they do is they actually take blood samples and run it through a test. So I'm wondering if you have plans to compare your device with some of the gold standards that are out there for measuring dehydration.
Oh yeah, absolutely. I actually didn't mention this when the slide came up, but on the slide where I talked about the market, the bullets mentioned expanding to athletics, but also other populations at risk of dehydration, like the elderly and infected patients that might be in the hospital. And you said that they normally do blood tests for testing dehydration. Those are incredibly expensive. And this might be a much more cost-effective way to get a simple reading initially, just to see maybe like how they're doing, how they're trending in terms of their hydration status. So that's a great question. Thank you.
Do you know of any other confounding factors that might cause the measurement of the saliva to be in error?
Yeah, sure. I mean, there are a couple. So one of them could be if the person just drank water recently, because some of the residual water in their mouth might affect the saliva sample. And so that's also part of the reason why we want to develop a more rigorous protocol. Part of the steps we want to implement is to restrict eating and drinking at least 10 to 20 minutes before samples are collected. Another confounding factor may be stimulation of the parasympathetic nervous system, I think, tends to give people a dry mouth. And so that's something we're also exploring. So there are definitely confounding factors. We're still doing the research to try and eliminate those while we do our next clinical trial. Any other questions?
Can you comment a little bit on cleaning your device? I'm just thinking of how grimy toothbrushes can get after a little bit of time, and now you've got people constantly using this, and then also in these field conditions that may not permit some sanitization of the device.
No, I mean, you're absolutely right. That was one of the things we thought about initially when we came up with the form factor of the saliva collection cup. And that's also why we're trying to transition our device to the like the in-mouth design, because that way we won't have something sort of just sitting in place, like saliva sort of like dripping everywhere. It's like, you're right, it's a little unsanitary. That was sort of just like an initial design that we had to do some initial testing. So yeah, that new form factor definitely won't have that problem.
So one last, look, we're almost out of time, but sorry to come back up. It struck me that you said,
no worries.
It struck me you said the 5-10K pathway that was the one you were going with a class 2 sort
Yep,
of
that's right.
approach. That seems a little over the top. Can you tell us about your justification about, for not pursuing class 1 sort of?
Well, sure. So, I mean, a lot of the commonly known class 1 devices are really simple things. So we're talking things like sterile gloves, tongue depressors, things like that would qualify as class 1. The other thing I should mention is that this device, especially in the new form that I presented up for something that's put in the mouth, we're pulsing electricity through those electrodes. And so it's a very small current, like not anything nearly high enough to actually damage or like injure someone. But just the fact that we're pulsing electricity through something that's in someone's mouth might cause our regulators to be a little sketched out, I guess. So we sort of want to be, conservative when we make that decision.
Thank you.
Okay, thank you very much.
Thank you all.
Our third presenting group is from Yale University.
Hi, everyone. We developed a novel platform for training healthcare providers on the use of external pacemakers to treat life-threatening cardiac events. So this is Maria. Maria was born with a variety of medical conditions that caused her to undergo open heart surgery shortly after birth. Maria's heart was connected via wires to an external pacemaker, which stimulates her heart in the case of an arrhythmia or abnormal heartbeat. Now, when the need for external pacemaking is required, the the trainers need to be trained in order to properly set the settings on the pacemaker to treat Maria. So Maria's case is not unique. Over 500,000 open heart surgeries are performed each year in the US, and over 40% of the post-operative patients experience arrhythmias that can require external pacing. And the external pacemaker is a complex device to utilize. And improper pacing results in serious adverse effects. Yet, no appropriate training platform currently exists. So, some of these arrhythmias that people experience are considered low frequency, but high risk. So that means that these nurses don't have the least amount of experience with the most dangerous arrhythmias. So one can see how simulation or practicing in these simulated environments can be beneficial in giving these healthcare providers the training that they need. So, despite tremendous advances in simulation technology, such as mannequins, such as you see on the screen, that can do everything from breathe even cry and sweat, there's no platform that trains on epicardial pacing. So we developed CardiSim, a responsive simulation platform for training healthcare providers in treating high-risk cardiovascular events. So before we understand how CardiSim works, we first understand how epicardial pacing works. So the pacemaker is connected to a patient via wires that are attached to a patient's heart and protrude out of a patient's chest. The pacemaker senses and listens to the patient's internal heart rhythm and then paces when it detects an arrhythmia. CardiSim simulates the patient. So CardiSim eliminates the need to have real patients for training. So it does this by interacting directly with an external pacemaker as well as a patient monitor. So here's how it works. So a trainer utilizes an iOS application that we developed to set the conditions of the simulated patient. So these include things like the heart rhythm, the arrhythmia threshold for capture. And then CardiSim, the CardiSim unit, produces an ECG, which you can see on a patient monitor. Next, the trainee utilizes the external pacemaker to try to correct the arrhythmia, in other words, treat the simulated patient. Now, the trainee receives real-time feedback because the patient monitor dynamically responds to the inputs that the trainee puts in the external pacemaker. So we took this concept, prototyped, and performed testing at the Yale New Haven Hospital with cardiologists, electrophysiologists, fellow nurses, and engineers. We tried to get in a wide array of people to utilize our prototype in order to validate our ideas and our device. Our device performed extremely well and we received lots of feedback, which we then incorporated into successive iterations of our device. So we believe that CardiSim uniquely satisfies customer requirements and market needs. So CardiSim is educational, it improves end care, it's easy to use, it's portable, and it's realistic. Perhaps most importantly, CardiSim is universal, meaning that it interacts with any pacemaker and any patient monitor. This is absolutely crucial because it means that nurses and doctors and other people who interact with the pacemaker are able to train on the exact equipment that they have in their hospital in order to provide the most realistic simulation possible. So given these benefits, we believe that CardiSim can capture the market. There are over 400,000 healthcare providers who will benefit right now from using our device. This is not just limited to cardiologists, but also nurses, electrophysiologists, anesthesiologists, et cetera. There are over 5,000 hospitals in the US, and 400 of those are considered teaching hospitals, where we would first target, because they would be able to incorporate our device into their teaching curriculum. Furthermore, since our device is a simulation platform, no FDA approval is necessary, resulting in a faster entry to market. Also, since there are no real competitors, we'll be able to capture the entirety of the market. Lastly, our chief physician mentor, director of the Yale Simulation Center, and is president of the International Pediatric Simulation Society, will be able to provide distribution channels given her contacts in the industry. So we've made significant progress so far. We are in conversations with the Yale Tech Transfer Office to best protect intellectual property. We received a grant from the Yale Center of Biomedical and Interventional Technology, and we're able to follow our budget appropriately to further technical development and perform market research. We presented at two, in addition to this one, we presented to two premier conferences on medical devices and BME. And most importantly, we have pending IRB-approved user trials in the Yale New Haven Hospital, which we hope to perform by the end of this year. It's been an ongoing collaboration with the Yale Engineering School, a fantastic cardiology team at the Yale New Haven Hospital, and the Yale Simulation Center, which have benefited greatly from their expertise in the industry. So given the investment the hospitals make in buying the pacemakers themselves and other simulation equipment, we believe CardioSim is well worth it. By improving education and end care and decreasing the duration, we believe that CardioSim is a cost-effective way to decrease the duration and costs of hospitalization, reduce complications, mitigate patient discomfort, and ultimately saves lives. Thank you.
Could you elaborate a little bit on what types of inputs are actually being presented to the person in simulation? Or is there a variety of different EKGs that are being displayed to them? Do they run through different scenarios? How does that simulation setup work?
Sure. Would it be possible to get the slides back up, please? So I believe your question was about, what are the various inputs that that the pacer, what can you learn on it? So we support a variety of arrhythmias, which you can see here. And there are a variety of concepts that can be taught, oversensing, undersensing, pacemakers. So like I said, the trainee would be able to set a condition of the simulated patient, and the trainee would be able to kind of learn how to treat the patient in this manner. Does that answer your question?
Can this only be used as an independent simulation, or can it be incorporated into the more comprehensive simulations for nurses to train with?
So one of the key aspects of our device, like I said, is that it's universal. So it interfaces with the exact equipment that the hospital has, but it can also be incorporated into the existing educational procedures. For example, if you're at a center that has a mannequin that does not support epicardial pacing, you can, for example, hide this, inside or a mannequin or somewhere. You could also use this in that sense as well. Yes.
Could you elaborate a little bit more on the potential market that you have? How many external pacing events or conditions are in a hospital or currently ongoing?
Right. So we said that there are 400,000 people who could be trained on this device currently. There are tens of thousands of pacemakers, external pacemakers that are currently in use. And we will be able to get people trained on that. Next question.
Hey, really nice idea. I work for Medtronic, used to work in the pacing group, so it seems really elegant. You surprised me when you said there is absolutely no touch point with a regulatory group, FDA to whoever. I'm thinking about laparoscopic surgery simulators. Are training tools regulated at all? And I'm sorry to ask this stupid question, but I didn't know that. It's not a class 1?
So I don't believe it's the case. Like we said, that the It's a simulation platform, so it never interacts with a patient. So this is only to be used with nurses, other people in the medical profession.
So to put on my FDA hat, if I give you a bad training tool and you learn poorly, does that hurt patients? Just throwing that out there. Maybe we don't need to debate this here, but it sounds like an IRB agrees with you, so it doesn't matter. But yeah, just
So we are to do user trials.
Yeah.
For that you do need an IRB.
Right, gotcha. Okay, thank you.
All right, thank you. Any other questions?
All right, thank you very much. Our third group is from Worcester Polytech. Fourth group, sorry.
Hi everyone, my name is Sneha Shastri. I'm from Worcester Polytechnic, and my project team created an automated system for high-throughput chemical screening in C. elegans. So according to the World Health Organization, neuropsychiatric disorders have the longest-lasting negative impact on those who suffer from them. Usually, these disorders, such as autism and schizophrenia, are treated with antipsychotics, and these medications cause really bad side effects such as muscle spasms, fatigue, and depression, and these are really obviously very bad. To take this one step further, 1 in every 5 US adults are negatively and directly affected by neuropsychiatric disorders. So these disorders, as you can see from this image, affect different regions of the brain. However, we can only measure this according to blood flow because this correlates with activity level. And we want to look at the neuronal level of this activity. However, in humans, we can't really measure this. This is unethical and impossible because the brain is such a huge circuit. So a good alternative is to use C. elegans. C. elegans are really small nematodes, and these worms are microscopic. Their connectomes are completely mapped, and they're intact, obviously. And there exist transgenic libraries where mutations that model human diseases can be replicated and easily implemented into experimental setups. So this is an example of an experimental setup. You would just pulse chemicals, and each column corresponds to a chemical pulse, and each row corresponds to a different worm. And we're looking at individual neurons in these worms, as you can see by the arrows labeled on different points on their bodies. So after testing what response the drug elicits, we can then see how each drug can modulate neuronal activity. And then to take this one step further, we can find dose responsing, and we can see what concentration can elicit the effective response that we want. So our lab has a manual method of conducting these experiments. It's very tedious and very time-consuming because we load worms into this microfluidic arena, and this arena is situated on top of a microscope, which is connected to a camera. However, the tube that connects to the inflow port is inserted into each chemical well, one after the other, manually by a person. And these plates can take up to 3 or 4 hours to test at a time. So as you can imagine, it gets very time-consuming having a person just sitting there, picking things up, putting them down. And so we wanted to automate this system. And we did this by creating a track system. So the X and Y axes are controlled, their movement is controlled by the sliders. As you can see on the X axis, there's an individual slider. And we wanted to attach this to the 96 well plate with all the chemicals in it. We also wanted to automate the Z-axis movement, which is the physical picking up and putting down of the tube. And this was a little bit less straightforward than the X and Y axes, which were just controlled by Arduino and stepper motors, so we had to go through a whole design process for it. Additionally, we had to create a platform and a sort of arm that held the 96 well plate away from the microfluidic arena and the imaging area so that no liquid spills would occur, because if you spill liquids on expensive microscope, that's a bad thing. So our design process for the platform was as follows. We designed things in SolidWorks, so the platform would just go right under the 96-well plate. The first iteration we made, we 3D printed it with rapid prototyping, and it broke. It snapped right in half. So our second iteration, we designed again in SolidWorks, and we did a finite element analysis. The maximum deformation in the bottom right corner was only 1 millimeter, which fulfilled our design constraint of 1 millimeter max deformation. And so we use this for our experimental use. Additionally, this is how the microfluidic arena and setup would go. So the plate would be under, and the z-axis motion had to be static. And it would just go down 5 centimeters and back up 5 centimeters, and then the plate would move from one well to another in half a second. And so for the z-axis motion, we considered several options, but we ended up deciding on the slide potentiometer. This option was really good for everything. Speed and cost was perfect. The only thing that was missing was that the control system was not direct position control. It was only motor speed control. So we had to design the direct position control system using Arduino. And we successfully did this. As you can see in this video, there's a top view and a side view of the system conducting the same user command. And so after we got everything moving the way we wanted it to, we conducted a cross-contamination validation test. So we did this using water and fluorescein, and we just tested 2 different methods. Our design constraint was that the maximum cross-contamination could only be 0.01%, and so we wanted to keep the level of fluorescein in water wells less than this amount. So with the first method, we found a cross-contamination of 0.025%, which was too high. And then the second method, with a rinse in between each test well, resulted in a sufficient cross-contamination percentage. So after we validated everything, we wanted to conduct a neural experiment and see if our system fulfilled what it was meant to do in the first place. So we replicated a dose-response curve, which is something that I showed you in previous slides. And it's very, very subtle, but if you can see, we're pulsing the worms with diacetyl, which increases the activity in the AWA neuron. And the neurons are slightly brighter right now, but I don't know if you can really tell. It's very small. But yeah. And so further research conducted by Ross Lagoy and Courtney Dick over the summer further confirmed our results. They replicated our dose-response curve as well, and it's the exact same thing. And then they also did a dose-response curve for NACL on the ASE neuron, which matched data from existing literature. And then they took this a step further and obtained data on optogenetic stimulation using our system. And if you want more info on this, they're actually presenting tomorrow, and they are poster 464. So to put this all into perspective, our lab wants to test 1,280 FDA-approved drugs, and we want to do this with 3 concentrations per drug. And this will result in 16 drugs per plate, and each plate will take about 3 hours, 15 minutes, give or take, to test. So total, this saves the researcher 242 hours of manual labor and just putting the tube in from one well to the next, which is also 30 full work days where they can do anything else as long as it doesn't have to do with drug screening. So I know there are existing products on the market similar to ours. After all, nothing is completely new. So I will say in response to that these systems are very, very expensive. And you might say, okay, well, you can just go to a different lab and sign up to use their facilities. But if you have a setup such as our lab does, where everything's very tightly packed and you can't really move things around, you can't just pick up your entire lab setup and put it into a machine like these. So our advantage is that, that not only is it cheap, it's just $300 for all the materials, it's also very modular. You can assemble this in any way that you want. We decided to assemble it in this shape and size, but you can make it however you want. So the conclusions we draw from this is that we fulfilled the design constraints and criteria that we had set earlier in the year. We then validated our system using a chemical cross-contamination experiment, and then we conducted C. elegans experiments on their neurons with the help of Ross and Courtney over the summer. And in conclusion, this is an inexpensive solution for drug discovery or honestly any other microfluidic application in which you need sequential liquid delivery to a microfluidic device. For our future recommendations, we would say that the platform could be made out of aluminum or some kind of metal to improve durability. We could also send an alert message to the user if an error occurs during the experiment and they're not right there to fix it immediately. And lastly, self-diagnostic correction of errors, if an error were to occur, would be ideal. Oops, sorry. So I would like to thank everyone who helped out with this project in any way possible. And here are a few reminders of what our system is and what it does. And at this time, I will take any questions from the audience.
Hi, I have two questions. First, who are your customers for this device? Is it research labs and pharmaceuticals companies?
Yes, correct. Anyone who's trying to use microfluidic devices for anything.
Sure.
Yeah.
And so at the cost point that you're at, how many of these do you think you need to sell in order to make a living?
I will go back. Hold on. Right. So our objective wasn't really on the profit and the patenting. It was more on the versatility of the product itself and its usage. So it only costs $300 for us, but we could easily just mark it up to maybe $500 and make a decent profit like that. And it would be a crazy competition for everyone else in the market because they're in tens of thousands, hundreds of thousands even.
All right, thanks.
Mm-hmm.
Do you have any plans to make this design freely available, or what would be your end game for it? I'm sure you've probably got some interest on your university even.
Yeah.
Hey, can we have one of those? But do you see this as just becoming freeware if you're going to publish on it?
So we were actually, we're still in the process of deciding whether or not to patent it or whether to keep it completely open source. Our entire report and all of the specifications are available on our school database. So they have the entire, diagrams and everything. But our main goal was to just increase usability and make sure that it's available for people.
You mentioned that it took 3 hours to fill one 96 well plate. So my question is, how long does it take to manually do it? Because I know when I do, for example, cytotoxicity studies, I can do a 96 well plate in 5 minutes manually. So to me, 3 hours seems excessively long.
Well, it's not to fill the plate, it's to test them. So we're running these liquids through a tube and then into a microfluidic arena, and we're measuring the response of the organisms in that microfluidic arena over a period of time. So that period of time when they're exposed to the chemicals accounts for the 3 hours. But the researcher has to sit there the entire time and make sure that everything's going according to plan.
So it's normally a 3-hour experiment either way?
Yeah, but this way it's just that no person is involved.
I had a question on your verification work that you did. Mm-hmm. You modified what you were doing to add a rinse step in there. Is that something that's typically done with other products on the market, or was it something particular with the way that you were doing it that required that step to be undertaken?
I believe it's something particular with us. Just from our manual experiments, we conducted experiments that way. So we decided it's ideal to just keep it the same methodology, but just take out the personal aspect of it.
Hi,
Would you say your device is error-prone to any specific step of the sampling?
Yeah, so in the earlier stages, it would kind of glitch out. We only used, Arduino and Micromanager, so it was a little iffy in the beginning, but the errors that occur now are very, very minimal, and we want to keep streamlining that and make them as minimal as possible.
Hi, can you only change the volume in the code, or can a user change the volumes they want to draw out on the outside of the device itself?
So right now, I think that's only controllable by the code. Everything is just conducted. You just press a button on the computer, and it just does the entire experiment for you. So maybe we could look into creating some kind of external device. Yeah.
Good.
That's a good idea. Thank you.
That's all we have. Thank you very much.
Thank you.
Our fifth group is from Virginia Tech.
Hello, everyone. We are the team from Virginia Tech. We are so excited to be here. I just want to do a quick introduction. My name is Catherine. This is Sarah. This is Nick. And this is Mike. And we are here to tell you about our device, which tracks real-time patient progress during a hemodialysis treatment using Raman spectroscopy. Okay, so 26 million Americans currently suffer from chronic kidney disease. 430,000 of those patients are currently on dialysis. So imagine being able to take a patient's satisfaction with their treatment from 50 to 90%. Imagine being able to just really increase their quality of life and provide them with an individualized treatment. Well, that is what our project goal has been from the beginning, and that will be the reality when our product is implemented. So as you can see from the graph here, the market potential for dialysis sector is pretty extensive, totaling around $30 billion a year. So I want to go over really quick a few of the problems with the way hemodialysis is currently done. Right now, there is really no quantifiable data of what is being removed from the body during a treatment. So a 100-pound woman and a 300-pound man, they get the same exact duration of treatment, the same exact treatment. So this lack of specification causes a number of side effects like, low blood pressure, fatigue, dehydration, nausea, and ultimately depression, because these patients have to spend so many times on their treatments, they really kind of do lack that quality of life. So our company name is Dialy Sensors, and the technology that really drives our company is called chemometric fingerprinting. So basically, it's a technology that uses Raman spectroscopy to kind of analyze a sample of a dialysis waste stream output, which creates a spectrum like you see on the PowerPoint here. So each small molecule is associated with a specific wavelength on this graph. As you see here, urea to the left, going over to glucose and then to water, and that's how you can see the intensity of the small molecules as they're being removed from the body. So this shows how we're really going to be able to make the personal care aspect of the hemodialysis process. This is just a urea peak, so we just chose one of the many small molecules that are removed from the body during this treatment. And as you can see, the peak goes, the intensity or the amount that's in the body obviously decreases as the treatment progresses. It's getting lower and lower as the dialyzer is removing it from the body. So a nurse or clinician would be able to look at this screen, or what it's on a computer screen right now, and be able to tell, okay, you've reached an acceptable level of the small molecule, we can now end your treatment. And that's how we'll really be able to incorporate the individualization.
Now, our team ran into a number of design challenges during the designing process. The first of which was that we had to isolate a static sample from a flowing stream to allow for better, more accurate Raman scattering. Then we had to design a sampling chamber which would reduce both the background interference to the Raman scattering and the contamination in the actual sampling chamber. Then we had to automate the actual device to enable consistent, more reliable patient monitoring over time. Finally, we had to make the design interchangeable between patients to increase the marketability of our design. and this all had to fit within size and price constraints. Mike's going to get into that a little bit more.
Yeah, so all these challenges shape and drive our design. So let me show you here what we have. Let me get the laser pointer out. We have a hemodialysis machine here that outputs a dialysis stream that contains all the solutes that are being extracted from your body during a hemodialysis treatment. So these include like urea, amino acids, glucose. So this is really good information that we want to tame. So how do we do this? We want to tame it with Raman spectroscopy. But we have a flowing fluid from here, from the hemodialysis machine, and we need a static sample. So how do we do this? We have a two-flow path system. We have the sampling flow path right here that proceeds this way, and then a bypass pathway to allow the sample to occur. This is connected by a three-way valve that directs fluid one way or the other. So while a scan is taking place, the fluid will go in the bypass pathway and allow for that Raman scan to occur. It takes about 30 seconds for that to happen. The good thing about this is after the scan is done, the valve redirects the new fluid and pushes out the old fluid so it's ready for another real-time scan. So we tossed around some ideas for the sampling chamber, and we wanted to get the best sampling chamber for the best Raman accuracy. So what we chose was an aluminum tube. We chose an aluminum tube because through testing we found that aluminum had the best reflective properties for an accurate Raman scan. So right here, this is the viewport for the Raman spectrometer to take a sample. So we also modeled this in ComSol, and so future thinking, we did find some dead zones that solutes might get trapped in, indicated by this blue zone right here. This is a velocity profile. So under future iterations of this device, we kind of want to smooth this out to make sure that there's no chance of solutes getting trapped at time one and then getting dislodged at a later time to affect with the results. So these are things we're thinking about. So we want our device to be as hands-off as possible. So we get some really good programming and soldering experience. What we did is we used a Arduino microcontroller to activate a relay to energize that three-way valve I was talking about earlier. So the program is set every 5 minutes we want to take a sample about during the course of their treatment. So it will energize the valve for 60 seconds to redirect the fluid around the sampling chamber. We do it 60 seconds to allow enough time for that 30-second Raman scan to occur. Now in the future, we want to connect all these devices, our sampling device, the Raman spectrometer, and also the hemodialysis machine so they can operate in sync efficiently in the future. Okay, so this is a picture of our first prototype. So we want to avoid cross-contamination between patients. We want this device to be able to be used on multiple patients. So we did that by the tubing set of our device is designed to be single-use and disposable. So every time, and in addition, the most expensive parts of our device, the pinch valve right here that we use, and the spectrometer, the Raman spectrometer act on the outside of the tubes and do not have any direct contact with the patient fluid. So every time a patient undergoes a treatment, they have a new set of tubes provided for them, so then their tubes can be hooked up to the hemodialysis machine, and then treatment can commence, and the Raman spectrometer can accurately analyze the solute concentrations throughout the course of the treatment and effectively increase the efficiency of hemodialysis.
Thank you. Over the course of this past year, we took our design from an idea to a working prototype. What you see up here is a cost analysis of this first iteration of working prototype. I want to draw your attention to the bottom, the cost of the Raman spectrometer. This cost is high. There's two reasons why this cost is high and will be lower in manufacturing. The first reason we expect to be much lower in manufacturing is that we'll be buying the Raman spectrometer in bulk. Whenever you're buying something in bulk, it automatically decreases the cost per unit. The second reason is, when we bought this Raman spectrometer, it was a full Raman spectrometer with every application that comes with the system. We only need a very few specific systems. When we incorporate the Raman spectrometer into our device with manufacturing, this cost will be cut dramatically because we will only need the applications of the Raman spectrometer for our specific device. Our bring-to-market price is estimated at $30,000 per unit, with a chemometric fingerprinting analysis scan approximating between $25 and $50 per scan, which would be used with our device. I want to walk you through now what our plan for commercialization is. So when a patient is suffering from chronic kidney disease, If they aren't having a transplant, they're undergoing dialysis treatments. These hemodialysis treatments are going to occur in a nephrology center or at home with a home hemodialysis machine, which is the exact same often that you would see in the nephrology center. As the treatment is occurring, our device will scan in real time the waste stream of the dialysate fluid that is flowing to drain. With this scan, an in-lab technician that is working with the hemodialysis center will be able to say, at this point, this person has hit the cutoff and can end their treatment. Another thing that it can do is the results will be sent to their personal doctor. With this, the doctor will be able to update and customize treatment plans, creating an individualized treatment for each hemodialysis patient. If you can create an individualized treatment, you can greatly increase their quality of life. If you're increasing the quality of life, one of the biggest things that we're going to see with that, shortened treatment times. Shortened treatment times means a nephrology center is getting through more patients. If you're getting through more patients every day, you're increasing revenue. There are a bunch of other ways that revenue will be increased with quality of life, but this is what we really want to focus on right now at this stage. So our revenue estimates over the first 2 years, if we're doing 5,000 samples at $25 to $50 a sample, that's $125,250,000 in projected estimated revenue. If we sell 10 analyzers in 2 years at $30,000 apiece, that is $300,000 in revenue just from the device alone. Now let's scale that up 5 years. 1.6 million samples, which is only 1% of the potential available sample population of patients and samples that could be taken on hemodialysis. That's a projected revenue of a minimal $41 million. Let's say we sell 1,000 analyzers. That's $30 million just in analyzers alone. Now, let's imagine. We take this 1% sample population as we sell more analyzers. Let's take it to 10%, to 20%. The revenue is only going to grow. Not only is the revenue growing, but the amount of people that you're changing lives, tens of thousands into the millions that are on hemodialysis currently, that are suffering chronic kidney disease, thousands that are on hemodialysis currently, you can change all of their lives with this simple device that we've created. We want to thank our advisors, Dr. Ryan Sanger, Dr. John Robertson, as well as Virginia Tech for its funding and allowing us to use its lab facility to create this device. We also want to thank BMES for allowing us to present our idea to you, and we want to open up the field for any questions that you guys may have.
Very good presentation as well. So my experience with readouts is they usually take a bit of time before you can get this continuous readout. So when you're developing a real-time device, how much of a delay is there? Can you quantify the delay before you can get these peaks?
Yeah, so actually we used a different Raman spectrometer in our initial trials that took, you're right, about 2.5 minutes for those peaks to be generated. But we actually discovered a new device called the Peak Seeker. It's made by Aldertron, and they're able to produce their peaks in about 30 seconds to a minute.
And a follow-up to that is usually when you get some results very fast, you don't know what the accuracy is. Can you talk about how accurate that reading is compared to what you would expect from a longer reading?
Yeah, those are definitely some experiments. We just got the Peak Seeker Aldertron that had that capability of scanning with the 30-second to 60-second time frame. So those are some further testing that we have to do to make sure that that accuracy is the same accuracy that we have with the previous Raman spectrometer that we used.
Okay, thank you.
I think this is a follow-up question to hers. When you did your testing of the prototype, what did you use for ground truth to measure against?
So we measured urine samples. So what we did was, so are you asking about the control with our device?
Go back to the slide with urea concentration versus time or something like that. I think it was charted on there.
Let's see. We'll get back to it here.
Go back a little more. There we go.
This, okay.
Yeah, so the y-axis on that plot is simply intensity. What's the quantitation of that?
Okay, so yeah, these are urine samples, and the intensity is correlated to the concentration, obviously. And so what we're looking for is a decrease in intensity. As we bring this to market, we could definitely correlate more directly the intensity to the concentration. once we get this implemented into the market. But right now, we're just looking at an intensity decrease or increase in solute concentrations as we scan these samples.
And how does that compare to current standard of care?
What we're seeing currently is a sample is taken through the blood. When a sample is taken through the blood, it is then analyzed. This is not in real time. What we expect now is that we can take this sample, we can run it through. You're actually going to be looking, as it's going through the filter, you should be seeing, right, a decrease in these small molecule concentrations. So what we see over time, it is a concentration, as he was saying. We're seeing that decrease, and we're able to take this measurement with a larger sample database. So if a patient is taking one sample, approximately once a month, maybe once every 2 weeks, to every time they're on treatment, every time they're in the dialysis center, every 5 minutes in the dialysis center, you're adding so much more data to their specific sample set that you can look more of what are some trends that you're seeing. How is their diet from last week affected it? How is their sleep affected it? How is their mood? Because these are all things that affect hemodialysis patients. If you can add more quantifiable data to this sample set, not only can you customize this treatment independently, but you can look at how are they clearing a specific biomolecule at a specific time point in the treatment.
Right, which is why I think it's important to make sure that your relative measurements here are correlated with actual quantitative values, because if you're going to shut off dialysis early on the basis of this, you got to make sure it's safe.
Right, and we actually have a grad student working with us who has normalized this data through multivariate analysis and compared that to what you've seen in a blood sample. And that is how we've created these peaks through that formula that he helped us develop.
All right, thanks.
Thank you. We appreciate your input.
One last question. Can you comment at all on the potential hemocompatibility of the use? I mean, you're basically stopping blood flow, doing an analysis on it in an aluminum chamber, and then letting that back into the bloodstream. Is there any potential risks there that might want to?
Thank you for the question. So what we're working on is the waste dialysis stream. So in the current hemodialysis machines, that's just it's under gravity. And so let me go back to this slide. Yeah, I know. Oh, went too far here. I'm going to go back to my first slide. So we are actually working on the opposite side of the blood side. We're working on the dialysate side. So that actually never goes back into the body. That goes to drain, typically. So that's like the information that's kind of going to waste that we want to capture. Does that answer your question? Thank you.
So I have a quick question. Based on your urea data, looking at the signal to noise, is the 0 minute and the 40 minute statistically different?
Yes.
Yeah, we are. We ran a few very, very simple statistical analysis on it. Can you go back? So it's really hard to tell here. Yes, you're looking at the intensity and you're saying that's not a huge difference, right? But it is when you're talking about a concentration of a fluid. So if you can go back one more slide. So here you're seeing this big urea peak. When you look at this urea peak compared to the glucose peak, that's pretty significantly different. So when we ran this through our analysis, we actually did see that, yes, while it looks like it's not that much change in concentration, the concentrations are already so minimal to begin with in the dialysate waste fluid stream that it does make a large impact. When a patient is on a treatment, you're looking to clear pretty small concentrations actually through the bloodstream. So as the blood runs through the dialyzer, cross counterflow with the dialysate fluid, which is what we're measuring, you're picking up these small molecules in very, very small concentrations. This small concentration is actually why we also have to use a stagnant flow. So the ramen can pick up these small concentrations, thus making it statistically significant.
Thank you.
Thank you very much.
Thank you.
And our last group is from Virginia Commonwealth University.
Good afternoon. First, we would like to thank the judges for allowing us to present our work here today. We really appreciate this opportunity. My name is Brittany Noah, and this is Brittany Martinez, and we're representing Virginia Commonwealth University and presenting our senior design project, the non-invasive blood glucose monitoring system under Dr. Paul Wetzel.
So the basic outline that we're going to go through this afternoon is we'll go through the background, market need for our device, a brief device description, how we looked at performance of the device, an economic plan, and then we'll have time for questions at the end. Okay, so the background. 383 million people had diabetes worldwide in 2013, and of this number, 3 million Americans have type 1 diabetes. So type 1 diabetes, diabetic patients actually have the occurrence of hypoglycemia, which is low blood sugar. And as time goes on, this can lead to further health problems as well as hypoglycemic unawareness, which is the inability to perceive those low blood sugars. So if this is undetected, these episodes can cause unconsciousness, coma, and death, and it's especially dangerous while they're sleeping because you have a reduced perception and awareness. So our objective was to develop a wearable nocturnal hypoglycemic detection and alert system for type 1 diabetics specifically in order to increase their peace of mind as well as their family's peace of mind and also reduce long-term effects of diabetes.
So now we're going to talk about the need for our product and the potential that it has in the market. So current solutions are either invasive or inaccurate, and there aren't any solutions that are purely alert devices. A lot of these administer drugs or some other type of preventive care, and we wanted to allow the patients to determine exactly what was best for them at that time through alert device. So our proposed solution was a non-invasive alert device that coupled heart rate with skin conductance, both of which have been determined by the American Diabetes Association to be symptoms of hypoglycemic episodes. So now I'm going to talk more about our device. This is a visual abstract of how we're collecting and analyzing the data. As you can see, we have two measurement streams, one with heart rate and one with our skin conductance sensors, and then it'll be analyzed to determine if the user is having a hypoglycemic episode. So now I'm going to talk more about how we're collecting and analyzing the heart rate data. So as you can see through the pictures on our slide, we're collecting ECG signals, which you can see at the top of each of the graphs, and then converting those signals into digital inputs for our lily pad. So we're filtering out all the peaks, all the other parts of the wave besides the R wave, which is the peak, and then calculating R to R intervals. So each time our lily pad, which is the microcontroller that we're using, receives a pulse, it is calculating that time from the last time that it received a pulse to get R to R interval time. This is a measure of heart rate because R to R intervals decrease as heart rate increases.
So now I'll talk about how we measured skin conductance in our design. So there are similar sweat glands in your fingertips as your armpits. So we utilize this information. So usually, you measure GSR through like a lie detector test via your fingertips. But we wanted to make our t-shirt ergonomic, so we didn't want anything on the fingertips. We wanted to embed our sensors within the t-shirt in the armpit. So what we did was we developed our own sensors. We have a layer of conductive fabric, a lead, and then we have neoprene, and that's sewn into the t-shirt. This is in order to increase, have a better signal and decrease noise. So with this, we can measure perspiration as perspiration increases, skin conductance increases, and that's measured through a differential voltage. So there's a sensor in the armpit and a reference sensor on the torso. So our final design looks something like this. We have the ECG signal and the conductance measurement. If both of those factors increase, that goes to an AND gate. If both of those are true, and the AND gate is also true, that alerts the user through vibrating motors embedded within the t-shirt to wake them up. So we used two different physiologic markers in order to increase accuracy and decrease false positives. So we'd like to introduce you to Doug. So Doug is our model of our prototype. You can see that he's wearing the microcontroller in the center. He has the GSR sensor, one in the armpit, one on the torso, and he has 3 ECG leads, 2 on the chest and 1 on the torso. We also have the vibrating motors down on the bottom of the abdomen. And we also have a hardware box that we utilized.
So now I'm going to talk to you about device performance through a video. Okay, so first we did a control at 60 beats per minute and a baseline galvanic skin response. And this is the heart rate simulator that we used. As you can see, it's set at 60 beats per minute. This is an usual skin response. And there's Doug wearing our t-shirt. And the vibrating motors are not going off right now. And then we did a second test at 120 beats per minute and increased skin conductance. And that's just showing 120 beats per minute.
Yep, they're vibrating.
And you can hear the vibration in the background. So in the future, we would like to streamline our electronics. We realize that our hardware box is not exactly ergonomic, but we would need the assistance of some electrical engineers and other people to simplify that. We also would like to increase the processing power of our microcontroller. We selected our microcontroller on the basis that it is wearable, not the basis that it is awesome at computing, but we would like some help in improving the computing power of it so that we can further analyze the data that we're getting. We've seen studies that have shown that spectral analysis of heart rate variability has been proven to correlate very highly with hypoglycemic episodes, and we would like to use this in the future instead of purely heart rate to have more accurate results. We'd also like to increase the battery life and make the device rechargeable, as currently it's using 2 watch batteries, which are inexpensive to replace, however inconvenient.
So now I'll go through the economic plan briefly. So we actually do have a provisional patent on our device. We made an initial budget of $750. We made our prototype for about $500. That's kind of overkill, but we had to order a lot of supplies initially. So we do know that FDA approvals and patenting are the next really expensive step in this process. In the future, we hope to keep the cost of production fairly low in order to only have the user pay maybe around $100 for commercial to pick it up in like the CVS or whatever. But if we do mass produce, like Brittany said, we would definitely need to streamline our electronics. We do need to keep in mind user-centered design should remain at the forefront, so we have to really think about safety precautions and protocols.
And we would like to thank our advisor, Dr. Paul Wetzel, his graduate student, Mr. George Weistroffer.
We would definitely like to thank the VCU BME department for funding our project and allowing us to be here today. And we definitely want to thank the Biomedical Engineering Society for choosing our project to present today. So thank you. We'll take questions now.
Over here. Hi, very nice talk. Maybe 2 questions if I could. One, did your market analysis consider that if this is as usable as you think, that maybe insurance companies would actually pick up the tab?
That's a really, really good point. So diabetes is super costly, as we all know. So this would actually help decrease cost for the insurance company, the user, allow them to not have as many future hospital visits potentially. But that's definitely a very, very good point.
And secondly, when you say increase in conductivity or increase in heartbeat, are there already thresholds out there in literature that you know what is the value of increase? That means a true statement?
There's some literature out there on it, but we would like to do our own studies to ensure that our device is picking up hypoglycemia accurately. And each person has their own definition of low, which makes it kind of difficult to set a standard across all type 1 diabetics. So that's something that we would like to allow for user input in the future.
Okay, thank you.
I have 2 questions. One from a practical standpoint of how do you wash said t-shirt?
Great awesome question. We were prepared for this. So all the sensors have snaps on the back. Can you hold the box? Yeah. So we brought it with us. All the sensors are snapped into the t-shirt so they can be snapped off and you can wash it. And there's a specific arrangement of each of the GSR sensors as well as the heart rate sensors, so the user knows where to put each back into, for sure.
And question two is, maybe this doesn't impact it or not, but if you've got somebody who gets really bad nightmares and dreams, I imagine their heart rate and sweating might increase. Is there a way to control for just a normal physiologic behavior versus a hypoglycemic event?
Right, so that's why we're interested in pursuing more complex analyses. If we did the heart rate variability spectral analysis, you would use Welch's method and do an averaging type filter on the data, and that would filter out that kind of false positive. But we thought about that a lot, and we decided that we would like to be woken up from bad nightmares, so we were okay with that.
Hi, I have two questions. One, some of the studies I've read have actually linked low blood sugar to a decreased heart rate. So how did you decide on measuring it with an increased heart rate?
We saw that information too, but the ADA suggested increased heart rate, and since they are the foremost on diabetes, we decided to go with their information.
And JDRF also said that too, which is a Juvenile Diabetes Research Foundation.
Okay, and then secondly, I'm type 1 diabetic, wear a continuous glucose monitor, and mine starts with a vibration, but I'm a very heavy sleeper, and the vibration actually never wakes me up, so it goes to like an alarm sound after a certain time.
Right.
Have you thought about incorporating something like that?
Yeah, we definitely have. I actually have a CGM too, I'm type 1. But I think that's a really good feature to have, and we've definitely thought about that. So with all of the software and hardware that we do have right now, that wasn't really in the works for us at that point, but I think it's something that we could definitely do. We had some really advanced ideas in the beginning of it with like apps and stuff like that, and like having an app for like parents in order to look at their children and stuff like that. So that's something that we definitely have thought about, and I think you bring up a really good point.
Thanks.
Thank you.
Hi, this is really cool, by the way.
Thank you.
As I think about your path from prototype to commercial product, the thing that strikes me is that you're going to need to make this thing really durable.
Yes.
So my question is, do you foresee having to modify your design for that, and will that impact cost?
So yes, we will most likely have to modify design. Our current prototype is very sensitive, shall we say. So we were
Did
gonna
you break it?
No.
Lots of
Not
electrical
yet.
tape, lots of electric tape. But we know that we're gonna have to embed the wiring within the shirt for sure, and that is definitely gonna impact manufacturing costs, because this is just a regular compression shirt from Walmart that was like $5, and that's gonna be way more expensive. But we're hoping that in mass production, since there are so many type 1 diabetics that could use this, that cost will be
And we
maintained.
looked into companies such as like Medtronic and Animas. The insurance company is a really good point. I feel like this is kind of like a global thing that could really impact diabetics in the future, and there's definitely going to have to be reiterations. Like there's always reiterations with medical devices, of course. So that's something that we could definitely look at.
All right, thanks.
Thank you.
If you plan on embedding the wiring in the shirt, is it still washable?
We could mess with that a little bit. So
it's definitely something that's been a priority for us.
Yeah.
That's why we did the snap arrangement a certain way and stuff. And you can look at it later if you're interested. But that's something that we'll have to deal with down the road in manufacturing. We haven't really gotten there yet.
we could potentially make the fabric specific to allow the wires to go up through it. Not necessarily within the fabric, but like make little loopholes so that way the person knows like where to put them. So they aren't necessarily like embedded within the t-shirt, but they're still embedded within the entire thing.
No loose wires.
Yeah.
Well, thank you very much.
Thank you.
So that draws to a close our design competition presentations. I'd like to thank all of the presenters again. You did an amazing job. Really appreciated it, and it was pretty exciting.